Welcome, everyone. Thanks for joining to the 2021 NeLLi Symposium. My name is Frederick Schulz, and I'm leading the New Lineages of Life team at the Joint Genome Institute. On behalf of the organizing committee, I'm excited to really welcome all of you to this first virtual NeLLi Symposium. In fact, it is the first time that we organize this meeting. The first one was in 2015 in Pacifica, California. The following two were coupled with the JGI user meeting. And now this year we have a slightly different format, which is a series of 6 online seminars that will take place every Thursday until October 21st. In these seminars, we will cover topics such as the evolution of the eukaryotic cell, latest updates on the tree of life, role of novel microbes in biogeochemical cycles, symbiont of microeukaryotes, and also latest developments towards methods that enable us to discover the currently undiscovered. In every seminar, there will be 2 main speakers with the talks followed by a short Q&A. Please post your question in the Q&A section during the webinar, and our moderator, Anya, she will then ask these questions directly to the speaker. Between the 2 main presentations, there will be a couple of shorter talks that we selected from the submitted abstracts, and there will be no Q&A after the short talks. And then in the end of today's session, we will have different breakout rooms in which you have the chance to meet the speakers, directly ask them questions, and discuss their research. Now, I will hand over to Anja Spang, who is a scientist at the Royal Netherlands Institute for Sea Research and who will moderate today's session. Anja, please take it from here.
Yeah, thank you very much, Frederick. Welcome everybody here. I want to introduce our first speaker of today, Laura Ehm. Laura received her PhD from Aix-Marseille University in 2011, after which she did a postdoc with Andrew Roger at Dalhousie University and subsequently joined Thijs Ettema's lab at Uppsala University at the time. That's also when we got to know and to work each other and got to work together. Since 2019, Laura is a group leader at the Paris-Saclay University in the DEAM team, and among others is supported by a ERC starting grant. So she has been a key player in the field of eukaryogenesis and driven many exciting projects on the origin and also the diversification of eukaryotes. And so today we are really looking forward to hear your talk on novel lineages of Asgard Archaeal and the insights they provide in the process of eukaryogenesis.
Thank you. So thank you very much, Anya, for the intro, and thank you to the organizers for the invitation. This looks like a great program, so I'm very excited. I'll start by, so hopefully you can see my screen. I'll start by just, before I present you this work, to thank the very many people that have been involved in this work and without who this could not have been possible. All of this work was done as my postdoc project in Thijs's lab, and I'm really highlighting the work of my 2 friends and collaborators, Dani Tamarit and Eva Caceres. So you'll hear a lot about this, and I'm excited to introduce the topic, but there's a long-going question about the origin of eukaryotes that has stemmed from the observation that eukaryotic cells really represent a chimera in the sense that they have features that are more similar to what you can find in bacteria, and they have other features that are more similar to what you can find in archaea. And of course, they have a number of specific features such as a complex intracellular organization. So today, I guess we will mostly hear about their connection and their similarity to archaea. And these similarities have been observed for a long time, basically since we've discovered Archaea. And the long-standing question has been whether eukaryotes represent a sister lineage to Archaea, which represents the three-domain tree of life here on the left, or whether they actually originated from inside Archaea, which is often referred to as the two-domain tree of life. It's been a long-standing question, and for many years, several decades actually, molecular phylogenetics have not been able to reach a consensus, and the analysis were often limited to a few genes that were available for the few sequenced lineages that were cultivated in the lab. And things changed quite dramatically with the advent of metagenomics, so the sampling and the obtention of genomic data from the environment. This has led us to a better representation of the true diversity of prokaryotes. And among others, so what has been kind of game-changing has been the discovery, so the publication of the first genomes of a new phylum of Archaea that were represented by this Loki Archaeota. Loki being a reference to where they were found. So those sediments were close to Loki's castle. And in the next couple of years, there's been the discovery of a few relatives of Loki Archaeota that have been altogether referred to as the Asgard superphylum. And why this was game-changing, among others, was that molecular phylogenies seemed to indicate that these represented the closest relatives to eukaryotes. So this was the situation in 2017, and you can see here that eukaryotes are represented as a sister group to Asgardarchaea, because it was somewhat unclear from the phylogenies whether eukaryotes were branching within Asgard or sister to them. And this is sort of the situation as I joined Thijs's group. And in addition to the molecular phylogenetics, there's a number of features, cellular features, genetic features that support this relationship between Asgards and eukaryotes. So what you often hear about referred to as eukaryotic signature proteins, or ESPs. So these proteins are proteins that are really important for the functioning, the proper functioning of eukaryotic cells. And for a long time, they were thought to be unique to eukaryotes. And as we discovered more and more archaeal lineages, we've discovered homologs of those proteins. And if you look, so here on the left is a very schematic tree of Archaea. And if you look at the distribution of those homologs in Archaea, without going in detail, you can see that there's a lot more homologs of those proteins in Asgardarchaea than in others Archaea. So this was an additional evidence of this specific relationship between Asgards and eukaryotes. Okay, so when I joined Thijs's group, our goal, we had access to I had access to a number of new Asgard genomes, and the goal was to refine this story. So to further investigate the phylogenetic relationships between Asgards and eukaryotes, and of course, to do some comparative genomics and better understand the genomic evolution of Asgardarchaea, to look for other ESPs if they existed, and investigate the metabolism of those lineages. Thanks to many collaborators, we had access to samples from all over the world, and we reconstructed 69 new Asgard genomes from different environments. And just very briefly, it's interesting to notice that, so this is just based on some representative genomes of different archaeal superphyla. And you can see that the Asgard genomes in general are quite a lot larger than most other Archaea. And without going into much detail, you can find them in very, very different environments, and they go from mesophilic to hyperthermophilic. And of course, so in the meantime, there's been a lot of work that's been done on Asgard-Archaeal. There's a ton of papers that came out revealing new lineages and investigating their ecology and their metabolism. This is only just a few of them. But long story short, so here are represented are in quotes new genomes, or 69 new genomes in red here, mapped onto a phylogeny of Asgard-Archaeal as was reconstructed as of August of this year. So what you can see is that we both have improved the taxonomic representation of some of the known phyla of Asgards, but we've also those MAGs, so those metagenomic assembled genomes, also represent some entirely new phyla. And I will not talk about all of them. However, I will just focus on one of them for the rest of this talk, which is the Eden Archaeota here. Okay, so one of the main questions that we want to answer is, how are eukaryotes related to Asgard-Archaeal, and are all the genomic data that's available today, does that help us precisely inferring this relationship? So the first thing we did was to use a data set that is quite standard by now, a data set of 56 ribosomal proteins. Those proteins are a standard when working at this evolutionary scale because they are slow evolving and they're universal. But they have also the downside of being all part of the same complex, which means that they can co-evolve. And I'll come back to that because this can sometimes lead to some biases. And they're also really short. So as you can see here with 56 proteins, you end up with around 6,000 amino acids to work with. So we developed an additional set of phylogenetic markers. So just to go to the point, we ended up with, we started from a list of markers that were identified to resolve the phylogeny of Archaeal. And out of these, we extracted the ones that had homologs in eukaryotes and were inherited in eukaryotes from Archaeal. And we ended up with a list of 57 new markers, and most of them are involved in informational processing. And as you can see here, so those markers are considerably larger than ribosomal proteins, since we ended up with about 14,000 amino acids. So we reconstructed our phylogenies based on those two data sets, and maybe you saw that coming, but we ended up with two apparently conflicting results. So the one good thing was that eukaryotes were, in both cases, really well nested within Asgard-Archaeal, which you can see here at the bottom. In one case, with the ribosomal proteins, they ended up sistered to Hemdahl-Archaeota, which was in agreement with what was suggested by previous analysis. Whereas in this new marker set, eukaryotes were branching sister to this one of these new lineages, the Eden Archaeota. So if we summarize this here, so this is what is represented here on the right-hand side. So in one case with the ribosomal proteins, the Eden Archaeota are going with the core Archaeota. So I should go back to this phylogeny here, sorry. So here in the ribosomal protein tree, the Eden Archaeota are not branching with the rest of the Asgard-Archaeal. Instead, they're branching in the sister group, so in the TAC Archaeota, and more specifically, sister to the core Archaeota. So this was a clearly conflicting result that we aim to better understand. And so for this, so if I summarize the different analysis we did, so we had two sets of markers, so the ribosomal proteins and this new marker set. We used both a maximum likelihood with IQ3 and Bayesian analysis with phylobase, and in both cases with complex mixture models. And we reconstructed phylogenies also by excluding some lineages. So by excluding, for example, the Archaeal, which are long branching lineages and that we know tends to distort the phylogenies. Same with eukaryotes and core Archaeota. And to this, we added additional treatment to the data. So we've investigated the effect of fast evolving site removal and recoding, and I will come back to that. Long story short, so we ended up doing a lot of phylogenies, and I will not obviously show all of this, but in short, the ribosomal protein data set came into agreement with the other marker set as soon as we removed the core Archaeota. So in this case, the Eden Archaeota go back inside the rest of Asgard, and more specifically, they branch with Archaeota and eukaryotes. And one of the main reason for this is that both core Archaeota and Eden Archaeota represent hyperthermophilic lineages. And what that means is you have a strong amino acid bias towards thermostable amino acids. This is a bias that exists across the entire proteome, but that is stronger in ribosomal proteins because they interact with tRNAs and mRNAs that are quite strongly affected by this adaptation to hyperthermophily. And so this was the main reason, this composition bias was the main reason for the attraction of Eden Archaeota to core Archaeota in the ribosomal protein dataset. And the placement of Eden as part of real Asgard Archaea was further supported by the fact that they encode what is considered to be truly Asgard unique eukaryotic signature proteins. So they encode bona fide actin and profilin, as well as a specific ribosomal protein that is only found in eukaryotes and in Asgards. So this is a story time that was, the idea was given to me by Danny Tamarit. So there's this story in the Norse mythology where Eden, who is the goddess of youth and belongs in the world of gods and goddesses, so in Asgard, gets tricked by a giant and is attracted outside of Asgard. And the gods, because she's the goddess of youth, the gods are very distraught by this because they start getting old and are looking for a way to bring her back. And so in our case, this mirrors our stories if we assume that this giant is the compositional bias in the long branches that are attracting Eden outside of core towards the core Archaeota. And the one way to try to bring back Eden to Asgard is to do proper phylogenomic investigations that we summarized here. So if in a very schematic way, if we start from our untreated data set, what happens is that eukaryotes branch with the Archaeota at maximum likelihood, and they branch deeper sister to Eden and Archaeota in Bayesian analysis. One of the treatments that we used that I've mentioned before is this fast site removal treatment. So in brief, you can sort the different sites in your alignment, so the different columns, the different amino acids, depending on their speed of evolution. And it's well known that fast sites tend to be poorly modeled, and they tend to lead to artifactual reconstructions, phylogenetic reconstructions. So we did an analysis where we removed more and more fast evolving sites. And when we do this, we do have eukaryotes branching deeper, so as sister to both Eden Archaeota and Hemdall Archaeota. Another treatment that is quite commonly used in phylogenomics is to use recoding. So recoding is basically, the idea is that, as its name suggests, is that you recode the amino acid sequence into a smaller set of characters. So what this means is that you're going to recode all the amino acids that have similar physicochemical properties into a single and identical state. And the reason to do this is several folds, but it helps, for example, with composition bias. So if you have some lineages that tend to prefer some amino acids over another, this is going to be sort of smoothened because of this recoding across different lineages. So this helps alleviating the artifact that can arise from composition bias. And so when we do this, in maximum likelihood, so eukaryotes tend to branch closer to Hemdall Archaeota, so they branch at the base of Hemdalls in maximum likelihood, and they actually branch within Hemdall Archaeota, and more specifically sister to one class of Hemdall Archaeota that we name Skadiarchaeial. And when we combine both of these treatments, then both methods will recover eukaryotes as branching sister to Skadiarchaeial. So as we submitted this paper, about 1 week later, this paper by the group of NeLLi and Eugene, who I'm sure will tell us more about this, came out. With a lot of new Asgard mags, a lot of very interesting genomic data. And as expected, the reviewers asked us to integrate this data into our analysis, which we did. And in very brief, we get the same result. This actually strengthens what we find in terms of the relationship between Skadiarchaeial and eukaryotes. I won't go into detail at all about the ESPs for the sake of time, but basically we searched for new eukaryotic signature proteins, which we identified and that are here highlighted in those colored squares. And among these, actually, quite a few of them are supporting this specific relationship between Skadiarchaeial and eukaryotes. And what's particularly interesting is a lot of them are involved in informational processing. Finally, something that we were very interested in doing was ancestral reconstruction. So you might have seen from those presence-absence plots that the ESP content across different Asgard lineages is very patchy across the different species. And it's the same for the predicted metabolism. And so we wanted to find a way to infer the ancestral characteristics of various Asgard ancestors. So for this, we used reconciliation methods, meaning that you're trying to reconcile the history of every single protein family onto the species tree. And you end up with something that looks like that, where you have on your species tree, you can map the gene losses, duplications, transfers, as well as the estimated proteome size of each ancestor. And one of the messages that we could take from those analysis was that the Hemdall Archaeota in general, and more specifically Skadi, tend to evolve by, they have undergone a lot more gene duplications than other Asgard lineages and than other Archaea in general. And they underwent less gene losses. And so this is somewhat reminiscent of what you can observe in eukaryotic evolution. So we can wonder if this was sort of the the predominant modus that started, like evolution modus that started evolving at that stage. And what's additionally interesting is that a lot of the duplicated gene content in Asgards include protein families that are involved in somewhat eukaryotic specific features, such as like cytoskeletal and membrane trafficking functions. And finally, we inferred that the Skadiarchaeial ancestor was quite large with over 4,000 proteins, whereas the last Asgard common ancestor was around 3,000. So as a take home, we have identified a lot of new Asgard mags, some of them representing new Asgard phyla. Our phylogenomic analysis do bring robust evidence for an Asgard origin of eukaryotes, and we think there's quite a strong evidence for eukaryotes being nested within Hemdalla-Kyoda, and more specifically as sister to Skadi. I haven't mentioned this much, but our reconciliation approach has allowed us to infer that the last Asgard ancestor of eukaryotes was likely a mesophilic heterotroph with a large genome and an increased gene duplication and decreased the gene loss rate. And finally, this has brought further evidence that complex eukaryotic pathways that are involved in protein targeting, membrane trafficking, and in genome maintenance and expression were inherited from the Asgard Archaeal ancestor. And thank you for your attention.
Thanks a lot, Laura, for your fascinating presentation. We can open for very few questions, maybe 2 questions. If you type them in the chat, and then we can take the other questions at the end. Maybe, and in the meantime, until there's a question there. I was curious, is the Skadiarchaeial group, does it include the LC3 lineage of Heimdall-Archaeota?
Yes, it does. Yeah,
which is one of the initial genomes. I don't see the Q. Okay, sorry, I was in the wrong chat. So there's a question from Carl Gras Gonzalez. Why is it so difficult to grow Asgard-Archaea in the lab?
Well, everyone would like to know that, I think. But, well, one of the hypotheses is that they probably need or possibly need specific symbionts, or at least other organisms in the medium from who maybe they get some specific metabolites. And so you would need to be able to maintain this community to be able to grow them properly. And we don't quite know yet what that would be, or who that would be, who the partner would be.
Yeah, this relates to a question of Alan Ko. What is the current metabolism and ecology of the Skadiarchaeial?
First of all, there still needs to be, because we're discovering their diversity more and more every day, and there's actually quite a few representatives now, we would need to know exactly what are the common traits. But they're mesophiles, they're heterotrophic. Actually, Anya has a paper with quite a detailed metabolic reconstruction of those organisms. They possibly need a symbiont.
Yeah, there are 2 other interesting questions, and maybe we can save them for the breakout session in the end, and so that we can follow the schedule with the short talks now. Thanks again, Laura, for your nice talk. And then we start the short flash talks with Burak Avci, who is a postdoc at the moment, also in the lab of Thijs Ittema, now at Wageningen University. And today he will present his work, which I think was recently submitted at bioRxiv, on fish microscopy applied to visualize Asgard-Archaea. So we are very much looking forward to your talk on the spatial separation of ribosomes and DNA in Asgard-Archaea cells.
Now I guess you can hear me, right? Yes, and you can also see my screen. Thank you, and thanks for coming to my talk. I will show you some pictures of Asgard-Archaea. These are the first pictures of Asgard-Archaea cell from marine environment, and I will talk about the separation of the ribosomes and DNA in Asgard-Archaea cell. And the big question in biology is the origin of the eukaryotes, also the theme of this session. And the recent data suggests that the merge between Asgard-Archaeon and alpha proteobacterium constitute the first cell of the eukaryotes. And the archaeal host is most likely Asgard-Archaeon, and then because we know that they are the closest known extant prokaryotic relatives of eukaryotes. That's what we know from the phylogenomics, and also Laura nicely summarizes the data supporting this. And also Asgard-Archaea genomes enrich in many eukaryotic signature proteins, such as for cytoskeleton or the membrane trafficking. And this all suggests that the subcellular complexity before the emergence of the first eukaryotic cell. So beyond these are the all genetic potential, but beyond the genomic potential, we need some microscopic evidence to demonstrate the cellular structure of uncultivated Asgard-Archaea in the environment. So seeing is believing. So to hunt Asgard-Archaea, we go for many sampling cruises in aerospace sediments. So I will just shortly summarize our workflow. We sample some sediment sample from aerospace, and we use a recently established workflow to get the full-length 16 sequence of Loki and Heimdall-Archaeota from aerospace sediments. So with this method, we don't use the primer. So this is a PCR-free method. So we can have we can capture the Asgard-Archaea diversity, and also we can design good probes to target most of the Loki and Heimdall-Archaeota in aerospace sediments. And then we label Loki and Heimdall cells in catalyzed reporter deposition fluorescence into the hybridization, so-called cardfish. And then finally, we visualize the Loki and Heimdall cells in the super resolution microscope. And the next slide, I will show you the super resolution microscope images of Loki and Heimdall cells. Green and red are the two fish probes with the distinct fluorochromes. These are the signals coming from the ribosome. We actually have these two probes to actually to see the true positive Loki and Heimdall cell and to eliminate the false negative cells. So this gives us a reliable selection of the Asgard cells. And also in blue, you see the DNA, the Dapli staining, and also here the overlay of the all signals. Here the scale bar is 1 micron. Firstly, they are in the coccus shape and in the size of 1 to 2 micron. And what we see, this is the first morphotype, is kind of half moon-like morphotype. The DNA is in the lateral position, and then we see the condensed and distinct fish signals. And the second morphotype is the ring-like signals. So here again, we see the fish signals coming from the ribosome as a ring, and also DNA signal is also condensed and separated from the DNA. And also here, there is also third morphotype. These are all some Loki and Heimdall cells will also form cluster you can also see the many Loci or Heimdall cells in the sediment. So this is the third morphotype. And again, we see the condensed and distinct DNA. And what we see, so this is actually what is fascinating of these pictures is the gap between the ribosome and DNA signal. So we can actually clearly image this gap thanks to the super resolution microscope. So, and if you take a section actually of the cells, you can clearly measure the gap between the ribosome and DNA signals. Here you see the fish signals in red and green, and also DAPI. And here for Heimdall and Loci, and then we can actually measure a gap from 50 to 280 micron between the DNA and the fish signals. And why it is unusual? Because in the prokaryotes, the ribosome and DNA or the DNA is dispersed in the cytoplasm, and then you see the partial or complete overlap of fish and DAPI signals. Here you see two examples from E. coli and also Taumarchion. Here, so you see the even DNA is condensed in particle points. You see the overlap of the fish and DAPI signals. So this is what we usually see. This shows us why this separated signal pattern with the gap is unique for Asgard cells. And this is my last slide, just to sum up. We do some research to answer some questions, but at the end, our answers actually made more questions. And so what we see, we observe the DNA is condensed and spatially distinct from the riboplasm. And what does it mean? It could indicate a kind of cellular compartment, or like planktomycete cell, it can be also the membrane, but in vagination to form a nucleoid region. And to answer this question, to find the proper answer, we need to see the ultrastructure of Asgard-Archaeal cell in the transmission electron microscopy. That's the future outlook. And actually our data is just being published in ISME journal last month. So for details, you can actually check the paper. And I want to thank all of the co-authors, all of the funding agencies, and also all people who help us for the lab and bioinformatics course. Thank you.
Yeah, thanks a lot for your nice talk. And we have little time, so we go directly to the next speaker, which is Yuzuki Hoshino, who is a geobiologist and currently works at as a postdoctoral researcher at the German Research Center in Potsdam. And today, he will present his work on the evolutionary origin of steroid biosynthesis in eukaryotes and implications for eukaryogenesis. So we are very much looking forward to your talk on tracing the origin of the eukaryotic cellular membrane.
Okay, thank you very much for the introduction. Yeah, so today's talk is about my recent work for the evolution of steroid biosynthesis and its relationship to the eukaryotic cellular membrane. Okay, steroids are one of three major lipid components in the eukaryotic cellular membrane, along with sphingolipids and glycerol phospholipids. And the steroid molecules are embedded in the eukaryotic membrane, and they play very important roles in the regulation of the eukaryotic membrane. And then, and the combination of these three major lipid components, glycerol phospholipid, sphingolipid, and steroids, is found only in eukaryotes. So the evolutionary origin of steroid is closely associated with the evolution of the eukaryotic cellular membrane. Eukaryotes are currently inferred to have evolved from Archaeal. However, the eukaryotic cellular membrane is fundamentally different from the Archaeal membrane, and little is known about how this transformation from the Archaeal type to the eukaryotic type occurred. So, for example, the Archaeal membrane is essentially isoprenoid-based phospholipid membrane, but the Archaeal type membrane is fatty acid-based phospholipid. And this is actually a common feature of the bacterial cellular membrane. So it can be hypothesized that bacteria may have may have contributed to the evolution of the eukaryotic cellular membrane. And the similar trend can be seen for steroids as well. So steroids belong to a large group of triterpenoids, and no cyclic triterpenoid has been discovered in Archaeal, but bacteria are known to produce a different type of triterpenoids called hopanoids. And both steroids and hopanoids are produced by homologous enzymes, and actually they also share a common biosynthesis pathway. So again, it can be hypothesized that bacteria may have contributed to the evolution of steroid biosynthesis in eukaryotes. This is a schematic diagram of steroid biosynthesis pathways comprising two major stages. So stage one is the production of a precursor molecule called squalene. And then the second stage is the cyclization of squalene, producing hopanoids and also steroids. And only steroid biosynthesis requires molecular oxygen. And in my current study, so comprehensive phylogenetic analysis of all of the steroid biosynthesis genes actually revealed that so there is an evolutionary connection between bacteria and eukaryotes. So there are two examples for the phylogenetic trees of stage one genes. So the tree topology indicates that these two stage one genes were horizontally transferred from bacteria to eukaryotes. And the same tree topology was indicated for the rest of stage one genes. And again, a similar tree topology was observed for the majority of stage two genes, including oxalosqualene cyclase, OSC. So this guy is responsible for the formation of steroid carbon structure. So again, eukaryotes in most cases clustered within bacteria. So altogether, 20 out of 26 steroid biosynthesis genes are inferred to be derived from bacteria. Especially this transfer was likely from myxobacteria. And this myxobacteria are aerobic group of bacteria and are evolutionarily close to anaerobic. They're the proteobacteria. So this observation actually is inconsistent with the syntrophic model of eukaryogenesis. So this picture is an example of modern syntrophic relationship between sulfur-reducing delta proteobacteria and also methane oxidizing Archaeal. And the syntrophic model proposes a similar relationship between delta proteobacteria and Archaeal at the beginning of eukaryogenesis. And my steroid data implies a long-term correlationship between delta proteobacteria and Archaeal. So on one hand, anaerobic ancestors of delta proteobacteria evolved into aerobic myxobacteria. And during this evolution, oxygen-sensitive stage one enzymes were replaced by oxygen-insensitive enzymes. And on the other hand, Archaeal ancestors also transformed into aerobic species by acquiring mitochondria. And then during this long-time relationship, steroid biosynthesis gene set is inferred to have been horizontally transferred from myxobacteria to Archaeal. And lastly, so steroid biosynthesis has an implication about the acquisition process of mitochondria. So there are two major hypotheses. So endocytic and also non-endocytic origins of mitochondria. So interestingly, if you consider the presence of steroids in eukaryotic cellular membrane, endocytic model causes a paradox about the origin of aerobiosis in eukaryotes. The endocytosis is a very complex cellular process and requires a dynamic membrane regulations represented by lipid rafts. And the lipid raft requires steroids and sphingolipids as major lipid components. And the steroid biosynthesis requires molecular oxygen. So this implies that proto-eukaryotes need to have been already aerobic before the acquisition of mitochondria. And this contradicts with the current assumption that mitochondria introduced aerobiosis into eukaryotes. So the steroid data implies that non-endocytic model is more plausible. Okay, so this is a summary, and thank you very much. This is the end of my presentation.
Yeah, thank you very much for your talk. And also here, I think we need to keep questions for the end to go right to the next speaker, which is Sori La. a PhD candidate in evolutionary biology at the University of Witwatersrand, and she will today present her work on the origin and death in the role of death in the rise of eukaryotes. The floor is yours.
Good morning. My name's Sori. I'd just like to thank you for letting me be a part of this conference and present my research titled The Role of Death in the Rise of Eukaryotes. So this is a diagram of eukaryogenesis, and it shows one of the commonly accepted hypotheses of the rise of feca through the endosymbiotic event. So it's expected that in this kind of a major transition, the symbiotic relationship would have constantly gone through cycles of cooperation, conflict, and conflict mediation for them to remain in a stable union. And it seems programmed cell death, PCD, played an essential role as a conflict mediator. In this talk, I'm not going to speak about why programmed cell death was so important for eukaryogenesis, but rather I'd like to talk about a part of my MSc project where I wanted to look at the evolutionary history of one of the most important effector genes of programmed cell death prior to eukaryogenesis and what this means. The genes I'm focusing on are the members of the C14 peptidase family, the homologs of caspases. So on the left here is a generic figure showing the molecular pathway of PCD. And the point I'm trying to make is that PCD is an extremely complicated process, but caspases are one of the main effectors in this pathway. On the right, there's a domain architecture diagram showing the caspases and the rest of the C14 peptidase members, and they're very well defined in terms of their domain architecture and that they're homologous. And in my study, I specifically looked at the homologs of caspases, the metacaspases and the orthocaspases, and their evolutionary history prior to eukaryogenesis. We won't be focusing on caspases or paracaspases at all because they're only present in eukaryotes. As I said, I am interested in where these PCD genes come from. And the two issues I wanted to address is one, the evolutionary history of these PCD genes in bacteria. And this has already been well established by Aravind and many other scientists who discovered the origin of eukaryotic caspases from bacteria, also called the bacterial connection. But the second issue I wanted to address, and that we actually don't know the answer of, is where did the genes in bacteria come from, and did Archaea have any of them? So the hypothesis that I'm interested in testing is that C14 peptidases are present in bacteria and Archaea, and two, C14 peptidases arose prior to the divergence of prokaryotes. Our approach was almost computational using classical approaches where I searched databases for Archaeal C14 peptidases, then confirmed them using hidden Markov model profiles. We also did searches of similarity and homology, as well as look at their evolutionary history. So I'd like to summarize the most important results. In reference to hypothesis 1, C14 peptidases are indeed present in both bacteria and Archaea. So on the schematic diagram of the three domains of life with Lucas, it shows that orthocaspases and metacaspases in orange and green are present in bacteria, eukarya, and Archaea, including the deep branching phyla as well. And the point of interest here is that although metacaspases and orthocaspases were found in these late branching archaeal superphyla and phyla, only orthocaspases were found in DPAN. And this distribution raised new questions and possibilities. Were these ancestral PCD effectors present prior to the diversification of Archaea and bacteria, basically Lucas, or did they emerge after their prokaryotic divergence followed by horizontal gene transfer? Therefore, we wanted to take this question further and test the second hypothesis. This is a gene tree of orthocaspases, and you can see that according to the different domains, according to different colors, are not clustered together. So basically, the gene tree didn't correspond to the species tree at all. And the most likely reason is extensive horizontal gene transfer. The fact that orthocaspases are present in deep branching Archaea and bacteria, we can actually show that one, orthocaspases are the most ancestral form of all C14 peptidases, And 2, presence of orthocaspase in all of the major lineages of archaea and bacteria suggests that they were present prior to the divergence of prokaryotes. While we've got evidence for HGT, we don't actually have proof that these ancestral orthocaspases were present since Lucas. And although we do agree that the tree is most fitting with HGT, we claim that presence of orthocaspases in organisms prior to prokaryotic diversification with HGT is the most parsimonious explanation. And this also fits in with Lucas or the forest of life concept. In conclusion, we identified C14 peptidases in Archaea, and we're quite confident that they were present prior to prokaryotic diversification. But what does this mean? If these C14 peptidases were present prior to eukaryogenesis, were they involved in programmed cell death, or were they involved in cell survival? This is because C14 peptidases are pleiotropic and they're involved in both cell death and cell survival pathways. Thus, we investigated the function of these ancestral orthocaspases by looking for death domains and other protein domains. We've progressed on this question, but for the sake of time, they'll be discussed at another opportunity and in future publications. Lastly, I'd just like to acknowledge these organizations for funding my research, as well as my co-supervisor, my supervisor. Thank you.
Thank you very much. We also have to move on very quickly because we run a bit late, and we go to Arkadiy Garber, who is a PhD student in the microbiology program at Arizona State University, working with John McCutcheon, and will today present her talk on regulatory adaptations during the early stages of endosymbiont genome decay. We're looking forward to hear your presentation.
Thank you. Thanks for the introduction, and thanks to the organizers for putting together this symposium and having me speak about my work. So today I'll be talking about some of our work looking into the early genomic and cell biological adaptations of bacterial endosymbionts. So the evolution of mitochondria is considered an important event for eukaryogenesis, and mitochondria came about through an association of an alpha proteobacteria within an archaea. And these interactions are thought to have begun billions of years ago, and much of the genomic signatures of the early stages of the interaction are now gone. So if we want to rewind the clock back and learn about what happened just after the interaction began, we can look at more recent examples of symbioses, particularly those involving bacteria that make residence within insects. So bacteria often make their way into eukaryotic cells for various reasons. Often these are pathogenic, but in some cases, long-term associations occur. And over long evolutionary timescales, bacterial endosymbionts undergo genomic reduction that causes them to lose up to 95% of their genome. And then this sort of model is sort of shown in this figure, where upon host restriction, a genome undergoes sort of a state of disarray with lots of rearrangements, many pseudogenes, a lot of gene loss, and this sort of continues until the genome is small and gene dense and looks like what we would call an organelle, like a mitochondria or plastid. So the McCutcheon lab is mostly focused on this right-hand end of the model, looking at the really small genomes and what they do. And my research, and in this talk, I kind of want to focus on what happens at the beginning, right after host restriction. So in the mealybug species that I study, P. longispinus, there are two young endosymbionts that very recently transitioned to an intracellular lifestyle. And they happen to live inside an ancient symbiont called Tremblaia. And Tremblaia are these light gray blobs here. And Tremblaia is an endosymbiont related to a beta proteobacteria, which itself houses these two young gamma proteobacterial endosymbionts. And this whole system is inside specialized insect cells. This is a really unique spatial organization. So given that these are young endosymbionts in this mealybug, we see genomic signatures consistent with the established model. So relative to their closest free-living relatives, these young endosymbionts have extremely high numbers of pseudogenes and really low coding densities. So pseudogenes are remnants of formerly functional genes, that have been inactivated, either due to some kind of disrupting mutation, or we see that the protein sequence of the gene is no longer conserved. So pseudogenes, the endosymbiont genomes are represented as circles here on the right, next to their closest free-living relatives, and pseudogenes are shown in red. So you see there's a whole lot of red here and not a whole lot of red on their free-living relatives. So with this information, we were curious about how this high state of genomic dilapidation is handled at the gene regulatory level. In other words, how has gene expression adapted, if at all, to the high density of broken and inactivated genes? So in both of the young endosymbionts, we see the pattern where expression of pseudogenes is significantly lower than expression from intact genes. And this makes sense at a broad level. However, these differences are not as extreme as we actually expected. So we see about 20% of the transcriptome in one of the endosymbionts still comes from pseudogenes, and in the other gamma proteobacterial endosymbiont, around 40% of the entire transcriptome is actually attributed to pseudogenes. So something that this was a surprising result for us, and led us to ask whether these pseudogene transcripts will eventually be translated into proteins, something that would be theoretically quite problematic for the cell, both in terms of energy wasted and potential toxic effects of the misfolded proteins. So subsequent mass spec analysis of these mealybug endosymbionts shows that the pseudogenes are actually not as well represented in the proteome. And those pseudogenes that are, those genes that are present in the proteome are shown here as blue dots. So we can see that from pseudogenes, there's actually not a lot of protein products being made, about 18 in the whole system, and they're at relatively low levels. So indeed, while pseudogenes represent about 20 to 40% of the transcriptome, they only represent about 2% of the proteome. So to summarize what we have so far, pseudogene formation is prevalent in these young endosymbionts, and gene expression seems to continue, implying that regulatory adaptation does not occur fast enough, or there's not enough selective pressure for it to evolve to outpace pseudogene formation and deletion. And the small size of operons in gene regulatory regions relative to coding regions also supports this. They are less likely to accumulate inactivating mutations fast enough to keep up with pseudogene formation in coding regions. And beyond transcription, it appears that pseudogenes do not leave much of a mark, and this is one of the ongoing questions we have in the lab. And how are pseudogene-derived protein products silenced? And one clue that we have about this comes from ribosomal binding sites and start codons, which we looked at on transcripts from pseudogenes and intact genes. And in both symbionts, we see this similar pattern where pseudogenes are actually less likely to encode a ribosomal binding site and a start site. So given the small mutational targets that ribosomal binding sites and start sites represent, it seems possible that these trends are under positive selection, where this endosymbionts are incentivized to silence pseudogene-derived transcripts, at least those that result in toxic proteins. But we still see quite a bit of gene expression from pseudogene transcripts that do have potential for translation, and this is one of the things that we're trying to figure out is how these are silenced. And with that, I'll say thank you for listening. Thanks to my lab, and yep.
Yeah, thank you very much, Arkadiy. We go right next to the last speaker of the short session, Sergio Muoz, who is a postdoc in the Dean team, also at Paris-Saclay University, like Laura, and has a major research interest in understanding the nature of the mitochondrial ancestor, and will today present a talk on the role of mitochondrial energetics in the origin and diversification of eukaryotes.
All right, can you hear me, Anya?
Yes, perfect.
Thank you very much for the introduction. And so I just want to say first that this is work that I did with Paul Schaefmacher, who is at Arizona State University. All right, so well, we all know that the origin of eukaryotes is a hotly debated topic. And one hypothesis that has become quite popular in the last 2 decades is the energetic hypothesis of Lane and Martin, which basically says that the origin of eukaryotic complexity requires a massive amount of energy. And one of the possible predictions of this hypothesis is that the energy demands of a cell scale discontinuously with cell volume. So we would see an energy gap between prokaryotes and eukaryotes. However, Lynch and Marinoff much more recently showed that the energy demands of a cell actually scale continuously with cell volume across prokaryotes and eukaryotes. But the problem with Lynch and Marinoff's interpretation and conclusions are that in their view, prokaryotes do not appear to be constrained by respiration at the cell surface. And that is in contrast with what we actually see. The data appears to suggest that there's an upper limit to the volume that prokaryotes can achieve. And so we focus on the respiratory membrane requirements of a cell and also the maximum volume possible that a cell can attain. And so we came up with this notion of the respiratory deficit, which is the ratio between the respiratory membrane needed by a cell and the maximum amount of respiratory membrane that is possible in a cell. And so if this ratio is larger than 1, a cell would not really be able to sustain its own volume by respiration at the cell surface. But if this ratio is below 1, then that cell can happily live by respiring at the cell surface. So we did some calculations and we applied this model. And we found that the maximum volume that a cell can attain depends mostly on 3 major parameters: cell division time, the fraction of respiratory membrane allowed, and the cell shape. So for example, if one were to increase the cell division time from 1 hour to 10 hours, the maximum cell volume that a cell can attain while respiring at the cell surface goes from about 1 cubic micrometer to 1,000 cubic micrometers. Then we also apply this model to data, to empirical data that comes from eukaryotes and prokaryotes. And what we find is that a lot of small eukaryotes really would be able to theoretically make a living by simply respiring at the cell surface. However, we also find that larger eukaryotes, as long as they have their own cell division times, the empirically determined cell division times, they do suffer a shortage of cell surface area. So in principle, mitochondria, they really derive a benefit from mitochondria. And then we focus on the energetic investments in DNA of cells with contrasting genomic designs. So this is an idea that Nick Lane introduced, and basically what he claims is that prokaryotes and eukaryotes scale up their genome numbers differently with cell volume. So for example, if a prokaryote were to increase in cell volume, it would also proportionally increase the number of genomes. But if a eukaryote were to increase in cell volume, then its nuclear genome doesn't really increase. What increases is the number of mitochondrial genomes. So then we can calculate the amount of energy that each one of these 2 different cells, prokaryotes and eukaryotes, are investing in DNA as a function of cell volume. And then we can compare that to estimate the possible advantage that the genomic design of eukaryotes has over that of prokaryotes. And so we applied the model, and what we found was indeed that prokaryotes progressively are investing more and more energy into DNA synthesis, and so they quickly run out of energy for other cell processes, but eukaryotes are really investing a negligible amount of their energy budget into DNA. However, we also find that a prokaryote would theoretically be able to reach a genome size of up to 30 megabases while only experiencing a 20% energetic disadvantage in relation to a comparable eukaryote. So to conclude now, we can say that prokaryotes do not really suffer a shortage of surface membrane at cell volumes before below 100 micrometers, cubic micrometers, and they only have a slight energetic disadvantage at a genome size of about 30 megabases. Extent prokaryotes and eukaryotes overlap at these cell volumes and genome sizes, and it has also been estimated that LECA had about 20,000 genes, and one could argue that possibly had a genome size of 20 or 50 megabases and a cell volume of 20 or 60 cubic micrometers. And I can give you the reasons for this later. And from these observations, then we conclude that the prokaryote-eukaryote transition may not really have may not have required mitochondria, but we also conclude based on our analysis that the successful diversification of eukaryotes was ultimately possible because of the origin of mitochondria, which allowed eukaryotes to eventually achieve much larger cell volumes and also faster cell division, faster growth rates. That's it. Thank you for listening.
Yeah, thank you very much, Sergio. That was a very interesting talk. And then we can move on to our last speaker of today, Eugene Koonin, which you probably all know, who is a NIH distinguished investigator heading the Evolutionary Genomics Research Group at NCBI since 1996. And he's addressing all kinds of fundamental questions in evolutionary biology using a large variety of approaches, including systems biology, comparative genomics, mathematical modeling, and so on. And of course, throughout his career, Eugene has really shaped the field of evolution, made many groundbreaking discoveries and wrote books, and has also, of course, been acknowledged with various honors and awards. So we are really happy to have him here today and look forward to your talk on entitled From Asgards to Eukaryotes and How Hard Was This Transition? Did you open your microphone? So far, we cannot hear you.
Now you can.
Yes.
But unfortunately, I have to restart screen sharing. My apologies. My apologies. In the meantime, while I'm doing this, I really want to thank the organizers for inviting me to speak at this exciting symposium, and Anya for her nice introduction. But I also have to point out, with apologies, that due to another urgent meeting, I had to miss Laura's talk this morning. So this, whatever I'm going to say, is exactly pre-Laura, without knowing what she actually told you. So Asgard, before today's morning talk by Laura, and the implications for the origin of eukaryotes. So one thing that I have to, can assume quite safely, though, that Laura gave you an excellent introduction to Asgard. And therefore, I can go through these slides very quickly. So it gives me special pleasure to mention this landmark breakthrough 2015 paper with Anya as the first author, where Asgard, at that time not even named like that, named just Loki Archaeota, after incidentally the most malicious member of the Asgard, has been discovered through rather heroic metagenomic analysis from Loki's castle in the sea bottom in the Arctic mid-ocean, and it has been shown in this study that the Lokis at the time, what we now know as Asgards, are the monophyletic group, and apparently a sister of the text superphylum of Archaeal, and lo and behold, in that work again, in the phylogenetic tree of a number of universal genes, universal phylogenetic markers, eukaryotes fall within the same branch did the Lokis. And on top of that, many eukaryotic signature proteins have been discovered, making the case that the ancestral parent of eukaryotes basically lies within this branch of Archaeal. And then in another groundbreaking paper a couple years later, also from Ty Sedema's lab, the Asgard have been substantially expanded. Several nucleates have been discovered, and additional eukaryotic signature proteins, and the group within the branch within Asgard, named after one of the deities, Heimdall, has been claimed as the closest archaeal relative of Eukaryotes, and this is where the when the word Asgard appeared for the first time. They have been really elusive in biological terms until last year, when through equally heroic efforts for quite many years in the lab of Avci, Italia, the first representative of Asgard has been shown. We can see them, and we can see remarkably that they lead syntrophic lifestyle with bacteria, which provides fuel to the syntrophic hypothesis of the origin of the eukaryotes, which have been developed by Puri Lopez Garcia and David Moreira earlier, and befit the ancestors of eukaryotes. Okay, so this is by way of introduction. And now a recent major expansion of Asgard and the implications for the origin of eukaryotes, which have been developed in our collaboration with the lab of NeLLi from Shenzhen University in China, published just recently. So what I'm going to discuss next comes mostly from this paper, and even more so from what remained behind the scenes in this paper. And I will hark back briefly to some earlier work to pat my own back towards the end of the talk. So what we had now in this work was 87 Asgard genomes that were available when we started, plus 75 new mags complete to different extent, amounting altogether to 162 distinct Asgard genomes. We certainly want more, as I will emphasize in a moment, but this is a respectable data set, I think. So our goals, our objectives in this work were several. First of all, it's sort of bioinformatic goal to create a platform, a framework to facilitate comparative genomic evolutionary analysis of Asgard compared to other organisms. So what we did to that effect was the usual thing we do, namely constructing clusters of orthologous genes, COGS for short, specifically for Asgard Archaeal, with considerable manual curation involved, primarily by Kira Makarova, resulting in nearly 15,000 of these COGS, and this is the important Asgard COGS, and this is the important number, the coverage. About 90% of the genes on average in Asgard genomes are covered by these curated clusters of genes. So basically, in a sense, this is a successful effort because we cover most of the genetic diversity of broad-scale genetic diversity of Asgard. Then the next set of tasks is phylogenetics, basically also twofold. First of all, we want to build trees to resolve the relationship within Asgard and to delineate putative new phyla. And then, of course, we cannot avoid it. We want to build the tree of life with the largest possible balanced taxonomic coverage of archaea, eukaryotes, and bacteria, primarily, of course, to resolve the Asgard eukaryotic relationship. And then, I would say most interestingly, we want to identify as many eukaryotic features as possible, but importantly not more, by applying the best of computational methods that are available to us in search of the ESTs. So to begin with, I will rather briefly cover the phylogenetics without dwelling too much on the phylogenetic debate, which is impossible to avoid when you talk about the deepest branchings in the tree of life. So how we did it briefly, for the Asgard tree, we had 162 Asgard genomes, and we sort of converged on the set of 209 phylogenetic markers, which are genes that are represented and reasonably well conserved in nearly all available Asgard genomes. You cannot realistically require absolutely all, because remember, these are megs, these are not closed genomes, so something may be missing, but nearly everywhere. And then for the construction of the tree of life, we have been more selective to construct a large but balanced set of genomes from the three domains of life, maybe to so 76 best covered Asgard genomes, nearly 300 genomes of other Archaea, 100 bacterial, 76 to equal this eukaryotic genomes, and they used the rather old and tried data set of universal genes from Per Bork's work, of which there were 30, one of which, and it's a sort of a warning tale, one of which they found to be unsuitable for this analysis with anomalous topology that, if used non-critically, swayed the tree towards the three-domain topology, which we now strongly believe is not correct. And they used the best available maximum likelihood approaches with the best feasible model, because the most powerful models are actually not practical for database, for data sets of this size. So what comes out of it? This is sort of just one composite slide where I try to present these phylogenetic results. First of all, the diversity of the Asgard superphylum itself. What we have here is the structure that is largely compatible with the previous phylogenies, but include 6 putative new phyla, 3 of which nicely group with the Heimdall Archaeota, sort of resulting in a expanded Heimdall super group, which is good, I would say, for trying to understand the origin of eukaryotes. And this is our main tree is based on concatenated highly conserved core proteins, but this is well corroborated by 16S RNA analysis. And I would like for a second emphasize this deep putative phylum, which is the deepest in the expanded Heimdall, and for naming which with our Chinese collaborators as a bit of an homage, we for the first time departed from the Norse mythology and named it Wukong, which is the Monkey King, sort of a Loki-like character in Chinese mythology. Now to the tree of life, which may be more controversial issue. In fact, then we built the tree from a concatenated alignment of 29 universal markers. We have Eukarya grouping not within Asgard, but deeply within Archaea, as a sister to the Tak Asgard branch. But when we do it in a somewhat more sophisticated way, namely that when we derive the consensus of bootstrap-like samples of these 29 markers, we do get the grouping of Eukarya reasonably well supported grouping of Eukarya with the expanded Heimdall Archaeota, which is the best we can achieve today, I believe, the most robust topology, although I would say that we need to investigate it further. We need more support. This case may not be considered clinched, although this is the most likely solution as far as I can tell today. Okay. Now let's look at the gene content of the Asgard, which in a sense is a more biologically meaningful, exciting subject. And what we see here is that when we cluster the Asgard genomes by patterns of genes presence, absence, using a PCA type analysis, we have actually rather nice clustering within this plane of principal components. Here is, for instance, Loki, here's Thor Archaeota, the expanded Heimdall supergroup with Wukong being somewhat peripheral. This suggests that at coarse grain, the patterns of gene gain and loss in the evolution of Asgard are compatible with the framework given by the tree topology or the universal genes. When we look at the functional breakdown of the core Asgard genes that are widespread in Asgard, compared to a similar breakdown for the tech superphylum of Archaea, what we see is an interesting picture in the sense that in most functional categories, in particular, most conspicuously in translation, of which there are many, universal genes. We have the lion's share of genes that are shared by Asgard and Tech and other divisions of Archaea. In other categories, we see a smattering of unique genes, unique to Asgard or Tech. But what is remarkable is that we see a large fraction of Asgard-specific genes in two categories: cytoscatal vesicular trafficking, intracellular trafficking, vesicular transport, and such processes, and energy metabolism. In this respect, Asgards are very distinct. More on this later. But also when we look at the evolutionary conservation of these genes, again, we see the greatest fraction of universal genes, a substantial fraction of genes that are common to Archaea and Eukaryotes, but not bacteria, and small fractions of, say, Asgard-specific genes. So things are, this is one of the main emphasis that I want to make. Things are not simple. All kinds of genes travel a lot across the diversity of life. And if we look in a bit of a different way at the expanded core genes of Asgard, we can see it all the more clear. Because here we just look by presence absence, yes or no. And what we see is that the majority of these genes are widely represented in Archaea, bacteria, and Eukaryotes. They're the largest, the next largest fraction are common to Archaea and Eukarya. Then those that are found in Archaea and bacteria, and there are very few clean signatures. those genes that we can detect in Asgard only, or perhaps this would seem more interesting in Asgard and only in Asgard and Eukaryotes. But unfortunately, there are virtually no such genes. So clean signature, clean, let us say, shared derived characters, synapomorphics of common origin between Asgard and Eukaryotes are nearly impossible to come up with. We need to go for more quantitative, more sophisticated analysis. And when we just do it by sequence similarity, by significant difference in sequence similarity, we see the same type of picture. Much of the time it is hard to tell by sequence similarity. Sometimes genes and many other genes are clearly archaeal. The rest are small fractions. Okay, so our next goal, perhaps the most interesting and most challenging, was to derive what would amount to a comprehensive set of Asgard ESPs, so at least as comprehensive as possible. In order to achieve that, we have been looking at all kinds of candidates, comparing to the previous best set in Theis's, in the paper from Theis's lab in 2017, comparing to all kinds of databases, and doing a whole lot of manual curation of potential candidates and the neighbors in Asgard genomes. So as a result of these many new ESPs not noticed previously were detected, we will mention in the next couple of minutes a few of them more specifically, but what I want to bring your attention to is this letter U, which comes from the COG functional classification and means this category of intracellular trafficking, secretion, membrane remodeling. So I would say overwhelmingly the ESPs, and in particular the newly discovered ESPs, are involved in this intracellular process. So, so far, among the nearly 15,000 of Asgard cogs, we have 5,000 of those that represent ESPs. Nearly half of them have not been previously detected, and strikingly, over 60% are of this secretion, intracellular trafficking, physical transport, functional class. So a significant majority. And we also see, very notably, dramatic proliferation of some key domains, which is in principle similar to what occurs in eukaryotes with very extensive serial duplication of some proteins and domains, such as the roadblock superfamily, such as small GTPases, and a few others. So when we look at this overall pattern or heat map of the ESP presence in different divisions of Asgard as color-coded here and here, the first and foremost thing that we see is the patchiness of that pattern. It's not really consistent. You always see vertically in terms of what you find in the particular group of Asgard, or horizontally in terms of where in Asgard you find this particular ESP. You see all these gaps in the patterns. Genes again come and go. But that's it. By far more consistent, most consistent, the only really consistent category of genes are again of ESPs in Asgard are indeed those involved in intracellular trafficking and related processes. And the expanded Heimdall group is indeed enriched in this category of genes as shown in this blow up, which is also patchy, but more consistent than elsewhere in the Heimdall. So evidently, we don't have a chance to analyze this sort of figures in detail. And for the rest of the talks, mostly, or my talks, these are mostly such figures. But what I want to impress on you is the high preponderance of multi-domain proteins among these ESPs. But the majority of them combine multiple ESP domains. And the remarkable observation that the majority of these domain architectures in Asgard are not the same as the domain architectures in Eukaryotes. The domains recur, the domains are the same or homologous, but the domain architectures are not. We shall talk about the possible reasons. About the last specific point that I want to emphasize is that the key protein complexes that in eukaryotes are essential for membrane biogenesis and trafficking, or at least multiple components of these proteins are indeed represented in Asgard. And I'll just rush quickly through these different complexes. Naturally, each of them carries an interesting story on itself, but we don't have the chance to analyze this. So very quickly, the Asgard complexes that are essential for membrane remodeling. You have most of the eukaryotic components represented by counterparts, by homologs in Asgard, with, again, unique architectures and complex operon organizations that tend to be combined in the same operon, emphasizing the functional connection. Similar situation is observed, let us say, with eufemylation, a distinct component of the ubiquity network involved in endoplasmic reticulum function, endoplasmic reticulum stress response, etc., which is distinct from other ubiquitin system genes, and so it is in Asgard. Because there is a variety, protein sorting is, which is essential for the functioning of eukaryotic cells, is accomplished by a variety of distinct protein complexes, such as, for instance, COP complexes, COP1 and COP2, the specific organization, and a variety of architectures of multi-domain proteins. And strikingly, within Asgard, we see all these domains of COP complexes, represented, but in domain architectures that are distinct from those in eukaryotes. The similar story is seen with the trap complexes that are interactors of COP complexes. Again, we see the counterparts of various subunits in Asgard combined in different ways. Now here's something interesting. The snare complexes, these are only represented in Heimdall genomes, being one of these, perhaps one of these rare synapomorphies uniting eukaryotes with their possible direct ancestors. And then this, the final thing, final specific thing I'm going to say, in the course of this work, we discovered the Asgard homologs of this DEN domain, which has a strange name, but in actuality it's a guanine nucleotide exchange factor for small GTPases of the RAB and RAG GTPases, which is a key component of the macromolecular complexes that are regulators of central metabolism in eukaryote combined with the mTOR kinase. The mTOR kinase is the central regulator of metabolism, which defines why some of us can eat all we like and remain slim and others do not. So the C, for the first time here, the counterparts or the main components of these complexes in Asgard. And in particular, the remarkable enrichment of proteins containing the DEN domain, bringing out, again, a variety of interesting domain architectures, bringing out the tantalizing possibility that the actually regulatory pathways of central metabolism, which in eukaryotes centered the mTOR kinase, are already exist in Asgard, although there is no homologue of TOR kinase itself, suggesting that a different enzymatic moiety is involved and challenging us to identify it. Okay, so some conclusions from this. Phylogeny, Archaeal, yeah, I probably should have added that the diversity of Asgard has been substantially expanded. Clearly counting, there will be more of putative phylum level lineages. I think this is not controversial. The Archaeal eukaryote relationship, our best take on this is that the direct ancestor of eukaryotes is a sister group of the expanded Heimdall supergroup. However, I think the case is not clear. Some trees point to deeper placement of eukaryotes and increased diversity, perhaps more lineages closer to eukaryotes should help, just like Asgard effectively solved the 3D versus 2D debate on the tree of life. Again, this is all said without listening to Laura's talk. Now, regarding the Asgard eukaryote relationships, Asgard are highly enriched ESPs. However, the pattern is not simple. Most of the eukaryotic signature proteins show patchy phyletic patterns. Many are scattered in other archaea as well. Asgard are just, so to speak, concentrate these, accumulate these eukaryote specific proteins, indicative of complex routes of evolution, extensive horizontal gene transfer. What is certain is the vast majority of ESPs in Asgard are related to intracellular trafficking, secretion, and vesicular transport, which clearly provides a link to the essential cellular functions of eukaryotes. Having said this, it must be noticed that most eukaryotic signature proteins, including those that are present in the majority of Asgard genomes, have domain architectures distinct from those in eukaryotes homologs, which sort of brings up two major possibilities. One is that these are not direct ancestors after all, and eventually we identify an archaeal group that will align with eukaryotic domain architectures in ESPs much better, or what I think is the preferable explanation is that there have been subsequent sort of parallel shuffling of these domain architectures in eukaryotes in Asgards. Some of the ESPs found in Heimdalls are even exclusively shared with eukaryotes, or at least more closely related to eukaryotic counterparts, and many ESP families are greatly expanded by paralogy, by serial duplication in Asgard genomes, which parallels what happens in eukaryotes. So, as I said, it's a very...
One more minute.
And I'm virtually done. As I said, I want to pat myself on the back a little bit at the end and return to some earlier work, pre-Asgard work, where we already, and others, of course, but I refer to our work, detected a variety of ESPs scattered around archaeal lineages and mapped their likely point of origin in the evolution of Archaea. I think this picture, this general pattern of gradual accumulation of eukaryote-specific proteins stands and just has been emphasized by Asgards. And again, this figure with the sort of conceptual scenario for the origin of eukaryotes comes from a pre-Asgard work and postulates the accumulation of complexity, the complexification of the archaeal cellular organization, the accumulation of different ESPs, particularly those involved in intracellular membrane functions. You could just as well write here Asgard, and that would sort of predict what happens. So basically, just for 1 second, returning to the finally to the question in my title, how hard it was to get from there, from the archaeal ancestors to eukaryotic cells. Very hard, but at the same time, there was the sort of pre-adaptation. A lot of the essential componentry has been already accumulated in archaeal evolution, and what it took, perhaps triggered by symbiogenesis, by acquisition of mitochondria, was sort of regrouping of these components to form the eukaryotic cell. I think it's a very general pattern in evolution that recapitulated at different stages. Origin of the eukaryotic cell remains elusive, but is getting less so with every new Asgard genome. I have to acknowledge participants in the work, Kira Makarova and Yuri Wolf, who did the bulk of genome analysis, as well as Anastasia Nikolsky and Natalia Yutin from my lab, who participated in some steps, and members of the Ming-Li's team from Shenzhen, from some other places in China, who really did the heavy lifting, the metagenomic analysis and assembly. I must mention this in the era of coronavirus. Here we are, Meng Li, Kira Makarova, and I, in 2 years, a little over 2 years ago, at the Gordon Research Conference at Les Diablerets, high up over Geneva, in Switzerland, getting together and conceiving of the work I told you about today. I think this meeting, all the Zoom notwithstanding, I think such meetings are indispensable, and we all meet physically soon. Thank you very much.
Thank you very much for this nice talk, Eugene. I suggest that we skip the questions because we are quite over time, and go directly to the breakout rooms, and then each speaker can have questions separately. And maybe Frederick, you will say a bit more, give more details.
just again, thanks to all the presenters for the really exciting talks. That was really great. And in particular, Anya, for sharing the fantastic session, everyone else who helped to organize the seminar series.